Assalamu alaikum dear students, hope all of you will be fine. Today we will discuss the physiological perspective of our digestive tract, and in the physiological perspective of the digestive tract we have to go through the study of the functions performed by the different structures or different anatomical structures which we discussed in our previous lectures of our digestive system. And the first most important anatomical structure of our digestive system is the mouth. And in our today's lecture we will come to know the role played by the mouth or the oral cavity or its accessory structures in the process of the digestion. So in our today's lecture we will be discussing digestion in mouth or digestion in our buccal cavity. Let us start. You know that the buccal cavity or the oral cavity or the mouth forms a specific zone of our digestive system in which or through which we incorporate the food items into our buccal cavity, then into our other digestive tube structures. So this very zone is called as ingressive zone. So the mouth or the buccal cavity forms the ingressive zone of our gastrointestinal tract, and we already came to know that the first part of our digestive system, that is the mouth or the buccal cavity, consists of so many structures, like it consists of the palate, soft palate, hard palate, vulla, tongue, teeth, gums, and it consists of the lingual frenulum, it consists of sulcus, which consists of sulcus terminalis, and so many other structures. And so far as the process of the digestion is concerned, we have to know that our buccal cavity has been provided with three important types of the accessory digestive glands, which are called as the salivary glands, among which the first one, that is the largest salivary gland, known as the parotid gland, which is present just lying below to our ears towards our interior side. In the infection, when mumps takes place in a person or in a child, then we can easily detect the location of the parotid gland from external side, because there is swelling from this and that side, isn't it? So the first and the largest form of the salivary gland is known as parotid gland, and the second one is lying just beneath our tongue, which are called as sublingual glands, and the third one is lying associated with our maxilla or this very and that very side of our buccal cavity, and they are accordingly called as submaxillary glands. And all the three types of the glands have their respective ducts, about which we came to know in our previous lecture, but let me remind you here that the duct which is belonging to the parotid gland, that very duct is called as Stenson's duct, and the duct which comes out from or which drains your sublingual gland, that very duct is known as duct of Ravenous, and the duct which drains your submaxillary gland, that is known as Watson's duct, isn't it? So this very gland, say, let me write down here, it is parotid gland PD, let me write down here it is sublingual gland, let me write down here it is submaxillary gland. Actually it is present towards the peripheral sides of our buccal cavity, but here in the two-dimensional board we can make the diagram only like this. And now let us have a look or the first performers of our different structures of our buccal cavity or the mouth, that is the teeth. Let us have a look over the role played by the teeth in the process of the digestive system. Teeth have an important role developed for the process of the digestive system, that is mechanical breakdown. That is mechanical breakdown of food. Once you incorporate different food items into your oral cavity, then quickly, if the food is solid format of the food, then you quickly are making use of your lower jaw, and your lower jaw continuously moves against the upper jaw, which is static in its position. Then the teeth which are present in our lower jaw, they get slided or they as if attack or push the teeth which are lying on our upper gums, and as a result of which the mechanical breakdown of the food takes place, isn't it? So the mechanical breakdown of the food takes place with the help of our teeth. You already know that our teeth are of different types. They may be incisors, canines, premolars and molars, and some are meant for biting, some for tearing, some for grinding, some for chewing. But the most important function among all these performed by our or the human teeth is grinding and chewing, because of which we can mechanically break down the large formats of the food items into the smaller items. And after that, what happens? There comes the role of these different types of the salivary glands. We have, parotid gland, submaxillary gland, sublingual gland. It will incorporate its secretion through which duct? Stenson's duct. Through Watson's duct? Through duct of the ravenous. And so far as parotid gland is concerned, it is mostly serous type. Its secretion is serous type, more watery, more fluid, isn't it? And so far as these submaxillary glands are concerned, their secretion mostly contains mucin. Henceforth, these maxillary glands contain mucus as well as serous. Their secretion is both mucus as well as serous. And so far as the secretion of sublingual gland is concerned, they mostly have mucus type of the secretion. This depends upon the presence of the type of the cells. This has mostly the goblet type of the cells, it has goblet type of the cells and the serous glandular cells, and it has mostly the serous glandular cells. Hence, their secretion is serous, their secretion is containing both mucus as well as serous fluid, and their secretion mostly contains the mucus fluid. And the secretion which is being secreted by all these three types of the glands in our buccal cavity is known as saliva. Under the influence of sympathetic and parasympathetic nerves which are located in our salivary central of the medulla oblongata of our brain. I will let you know about that in the later part of my lecture. First, let us know that the secretion which is being secreted by these three types of the glands, that secretion is collectively called as saliva. Now, this saliva is digestive in function and it has so many other functions as well, but let us have a look first over the composition of the saliva. What is saliva? It is the secretion of all these three types of, it is this watery secretion of all these three types of glands, isn't it? Now, if we will be having a look over the composition of the saliva, then we can easily find that it consists of water. It mostly consists of, let me make use of another color here first. It mostly consists of water. Then it consists of organic substances as well. It consists of organic substances. I will let you know which types of the organic substances are present in the saliva, and it also consists of inorganic minerals, or simply let me write down here, it also consists of minerals, isn't it? So far as the content of the water in the saliva is concerned, the presence of water in the saliva is more than 95.5%. Then a least percentage of other things remains there. But most important function is for the process of the digestion takes place because of the organic substances. And so far as the organic substances of the saliva are concerned, the organic substances are present in the form of enzymes. These are the important organic substances. And so far as enzymes of the saliva are concerned, we have different types of the enzymes. The first type of this very enzyme is known as salivary amylase, which is also known as ptyalin, Then there is second type of enzyme known as lysozyme. We have another organic substance which is present here in the saliva, that is mucin. So far as organic substances of the saliva are concerned, we have mucin, we have lysozyme, we have salivary amylase or the ptyalin, the functions of which will be discussed right now. And besides that, we also have some minerals present in our saliva, and minerals contain the inorganic chemicals like chlorine is there, like potassium is there, like sodium is there, like thiocyanate is there, like carbonic ion is there, etc. These are some of the ionic formats which are present in the form of the minerals in our saliva. So, saliva contains inorganic water, it contains organic substances in the form of the enzymes, and the mucin enzymes may be in the form of one important that is the salivary amylase or ptyalin, and it also contains another type of the enzyme, that is lysozyme. Here you should remember one thing, that the salivary amylase in we people is present in its two formats. Number one is known as alpha salivary amylase. I will write down here only alpha type, and number second is beta salivary amylase, or I will write down here beta type. Our salivary amylase is present in two formats, one is alpha amylase and another is beta amylase. So far as predominance among these two is concerned, alpha amylase is most predominant in comparison to the beta amylase of our saliva. Hope you are getting my lecture well. And now, so far as other characters of our saliva is concerned, let us have a look over some of the physical characters of saliva, and I will make use of another color for that. Please have a look for some physical characters. So, physical and chemical characters of the saliva. Our saliva has a specific gravity ranging from 1.002 to almost 1.008. This is the range of the specific gravity of our saliva, isn't it? And so far as it is chemical property, that is pH is concerned, it is pH ranges from 5.6 to 7.6 with an average of 6.8. Isn't it? Please remember these two things: specific gravity of 1.002 to 1.008 and pH ranging from 5.6 to 7.6 with an average of 6.8. These are the points which are related with the secretion of our salivary glands, isn't it? Now, let us have a look over the role played by these different types of the enzymes. Once our food is being chewed by our dentition or by our teeth, then the role of alpha type and beta type of the salivary amylase as well as the lysozyme comes. See what happens. It is this alpha and the beta amylase which are responsible for conversion of some carbohydrates like starch is there, isn't it? They have the property to break a specific binding, mostly alpha 1,4 linkage. You know, in most of the carbohydrates, the monomer units are binded with one another with respect to alpha 1,4 linkage, and that very bond is called as glycosidic bond. And the role of salivary amylase or the ptyalin comes here when we take carbohydrate rich food like starch. In the starch, the monomer units are connected with one another with the help of the alpha 1,4 linkage, and it is our this salivary amylase which breaks that linkage. With the breakage of that linkage, this starch is biochemically dissolved and there is a release of some lower forms of the starch in which maltose, isomaltose, dextrin like substances are there. We will get then here isomaltose, we will get maltose, we will get dextrin, isn't it? Wait, and let me check whether my phone is recording the lecture or not. Yes, and there is also release of glucose. You know, these are some of the monomer units of the larger carbohydrates. So, what our saliva has done here, in our saliva, the presence of salivary amylase, alpha type, beta type, it will break the beta linkage, it will break the alpha linkage. This salivary amylase has resulted in the biochemical breakdown or digestion of larger these carbohydrate units like starch, and it has led to the formation of these substances like isomaltose, like maltose, like dextrin, like glucose, with the breakage of the glycosidic bond present in the starch like carbohydrates. And then we should know the role played by the lysozyme. Lysozyme has the ability to break larger polysaccharide units, larger and complicated polysaccharide units. Why? You know, once you are taking food, along with the food you think that you have taken water, you have taken eatables like salad is there, like beef is there, mutton is there, chicken is there, but you don't know that along with that types of the food items, you are inculcating so many bacteria with your food item, and such bacteria which get inculcated through this ingressing process of our digestive system, such bacteria are being neutralized by the lysozyme, because lysozyme has the ability to break the complicated larger polysaccharides which form the cell wall of many bacteria. So many bacteria are there whose cell wall has complicated polysaccharides, and all such polysaccharides are being mostly neutralized or dissolved by our lysozymes. So, our saliva will be definitely having the antiseptic activity. We can make use of saliva for antiseptic purposes. See, sometimes if my skin will be infected and I will develop ascites, if some specific forms of bacterial infections are there, then I can make use of saliva and I can use that saliva in absence of any medicine as an antiseptic remedy against some bacteria, against some superficial skin infections. This is the God given gift, isn't it? So, after this, we also have different minerals, and we have also water in our saliva, and there is role of chlorine, potassium, sodium, which are maintaining the pH and specific gravity of our saliva. So far as the pH is concerned, we should round in from 5.6 to 7.6. In this range, our salivary amylase and our lysozyme like enzymes act optimally. In order to maintain that pH, there is presence of all these ionic substances or ionic minerals, and it is because of the prevalence of the water, which has a good role for the maintenance of the pH, for the maintenance of the specific gravity, for the maintenance of physical characters, for the maintenance of chemical characters of our saliva. If we will see on daily basis, if we are eating different things, different food items, then on daily basis we are releasing up to 1.5 liters of saliva per day. An average adult man, while he takes food normally 2-3 times and other things and eatables, fruits, etc., on an average he releases 1.5 liters of saliva daily. Sometimes for biotechnical analysis, we give some specific types of the chewable substances to the clients, to the persons with which there is more release of saliva. And let me write down here a name, that is paraffin. Sometimes we ask a person to chew paraffin, that leads to release of more and more saliva, and with the release of more and more saliva we can take his or her saliva for the biotechnological or biotechnical analysis. After that, let us have a look over the control or the nervous control of the secretion of our these type of the digestive secretion. So let us write down here. Control of salivary secretion. Control. So far as this reflex arc is concerned, reflex arc, it is our 7th and 9th cranial nerve. Remember please, 7th and 9th cranial nerve, which are associated with the regulation of the secretion of these 3 formats of the salivary glands, isn't it? But so far as the nervous center of our salivary glands are concerned, it is lying in our medulla oblongata. See, this is the medulla oblongata, and here we have salivary center, and our salivary center, it has 2 components to regulate the synthesis of saliva. One component is sympathetic and another component is parasympathetic. We know it controls the activities of the different structures through sympathetic nerves as well as parasympathetic nerves, isn't it? And here when we are having a look over the medulla oblongatory salivary center, we are finding 2 of its respective regions. One is controlling the salivary secretion through its sympathetic nerves, and another is controlling through its parasympathetic nerves. And parasympathetic nerves are primarily responsible for increase in the secretion of saliva, but that saliva contains less organic substances. Increase in saliva, but remember here one thing, when our salivary central regulates the synthesis of the saliva through its parasympathetic neurons or parasympathetic nerves, it leads to increase in saliva, but that saliva contains less organic substances. Remember please, less organic substances. So for example, I will tell you that today I am going to serve you a very good feast, I will serve you the lemon water, I will serve you the chicken, I will serve you the mutton, I will serve you the bath, and you will think about it. Once you will think about it, then your respective center through its parasympathetic network will induce your salivary secretion, and your mouth will get more of its saliva into its cavity. But you will say me that our mouth has watered, and if we will be having a check over the composition of that very saliva which has been synthesized or secreted after you have thought about an eatable, after you are thinking about a particular eatable, particular feast, isn't it? Then that saliva is containing less organic substances, isn't it? Now, if I will serve you, I will not induce thoughts in your brain, no, but I will serve you a feast. I will serve you the lemon water, and you will see it, you will physically touch it, and you will take it, and you will incorporate it into your oral cavity. Your teeth will begin to chew it, convert it as larger components into smaller components. Then your respective salivary center of the medulla oblongata will be making use of these sympathetic nerves, and at that time what will happen? There will be increase in secretion of what? Increase in the secretion of saliva, and that will be more organic. More organic. Why so? Because now you are taking the food physically, and your food might contain starch-like substances. Now your food needs to be digested, or the process of digestion needs to be started from your oral cavity. So for that purpose, these sympathetic nerves play an important role, isn't it? So far as the control of your salivary secretion is concerned, your salivary secretion control is lying in your medulla oblongata, which controls or regulates the secretion of your salivary glands through sympathetic regions or parasympathetic regions. And when parasympathetic nerves are getting activated, they result in the increase in the secretion of the saliva, which has less organic substances, isn't it? And when your salivary center induces your salivary glands through their sympathetic nerves, then that leads to the increase in the secretion of the saliva, which contains more organic substances. And you know your organic substances contain mucin, lysozymes, salivary amylase, alpha type, beta type, isn't it? I hope that you got a good understanding of the salivary center control and its regulation. And so far as the reflex arc in which your brain is also associated, then I have already told you that it is your 7th and 9th cranial nerve which is actually responsible, or which is actually associated with that respective reflex arc which is associated with the regulation of the salivary gland secretion. This is the main role of our buccal cavity or its respective accessory digestive glands in the process of the digestion. But we have seen that in our buccal cavity, both mechanical as well as biochemical breakdown of the substances takes place. Besides that, so far as our saliva is concerned, we came to know that it contains also substances. When it has been seen, when we are chewing a food item for more than a minute, for more than 30-40 seconds, then that leads to dissolution of the food item with more and more saliva. And it has been also seen that once we incorporate or swallowing process takes place, when incorporate the food or the bolus into our stomach, then for at least half an hour or more than 20 minutes, the salivary amylase which was mixed here in the buccal cavity with the food item, that acts over the breakage of the alpha 1,4 linkage of different polysaccharide units. So it is a good advice for every person that once you take solid food, especially when it contains the carbohydrate rich food, he should take some time to chew the food particles to swallow it. Isn't it? Once he will take more time, then more of the saliva will be mixed with his food item, with his bolus, and that will increase the efficiency of the digestion of that very person. That will increase the efficiency of the enzymes which are present in different other parts of our digestive tract for the digestion of carbs or other biomolecules. Isn't it? Now, let us have a general look over our today's lecture, in which we came to know about the process of the digestion in our buccal cavity, and we have seen that the process of the digestion of our buccal cavity, two types of the breakdowns takes place. One is mechanical, in which our teeth are being used, and another is biochemical, in which our enzymes are being used, which are present in the secretion of these salivary glands. And we have seen three types of the salivary glands, that is parotid, sublingual, and maxillary glands, and their collective secretion is called as saliva. And we have seen the physical and chemical characteristic features of the saliva and its composition. We have seen it contains most of the water, it contains organic substances, inorganic substances. So far as organic substances are concerned, it contains mucin, it contains lysozymes, it contains amylase, which is a two types, alpha type and the beta type, and the most predominant among the two is the alpha amylase type, and it is this alpha amylase which is responsible for the degradation or for break up of the alpha 1 for linkage of many carbohydrate units like that of the starch, and when it acts upon the starch like substances, it results in the release of the isomaltose, of the maltose, of the dextrin, of the glucose, etc. One more thing I have missed here, that is, if you will be served a biscuit, or if you will be served a food particle which is containing carbohydrates, and if you chew it, you will feel sweet taste in it. If you chew it more, the more sweeter it will become. If you chew it more, the more and more sweeter it will become. That is primarily because of this, that your more and more alpha amylase or the salivary amylase is getting combined with the food item. It is degrading the alpha 1 for linkage, that is leading to the release of the glucose, and you know the taste of the glucose is sweet, so your eatable is becoming more and more sweeter once you are chewing it, once you are masticating it continuously. And we have also seen that in an average person there is release of at least 1.5 liters of the saliva on daily basis if he is on his normal diet. So far as the control of the salivary glands are concerned, we have seen that the reflex arc which is associated with this activity is, in that reflex arc it is the 7th and the 9th cranial nerve which has its association, and so far as the regulatory control of these glands are concerned, the regulatory center for these glands are present in the medulla oblongata, and the salivary center of the medulla oblongata is performing these regulatory functions through the parasympathetic nerves and sympathetic nerves. Through its sympathetic system, it results in the increase in the salivary secretion, but that very salivary secretion will be containing more and more organic substances used in the process of the digestion. And when parasympathetic nerves are being utilized by the respective center for the secretion of these glands, then that very secretion will be containing saliva but less in its organic substances. And one more thing, let me take here, that it is not necessary that every type of the animal will be containing salivary amylase, alpha type, beta type. If we will take the example of these dogs, they don't contain these salivary amylase up to that extent in which it is present in the human beings. Or if we will take the example of the sheep, their saliva is not also containing such type of such quantity of the salivary amylase. In that we may find another less functional enzyme, that is the catalase, but Almighty Allah has provided us salivary amylase of both of its types in our saliva. So it is good for all of us that we should masticate well of our food. Then if you will be asked what are the other functional perspectives of our saliva, it is the saliva which is responsible for keeping your mouth It is the saliva which is responsible for keeping your mouth clean, and it is this saliva which is responsible for the provision of the resistive functions or the immunity functions against most of the types of the pathogens. So it is defensive as well. Your saliva is defensive, meant for the defense as well, and it also keeps your lips, isn't it, your tongue, your teeth moist. This is another function, and it is this saliva which helps you for ingression process of your food, for deglutition process of your food, in which you incorporate this bolus deep into your pharyngeal region, then deep up to your esophagus. So it helps us in the process of deglutition. It helps in the process of deglutition. It also acts as I already told you, antiseptic secretion, because it also has presence of another organic substance like immunoglobulins, and immunoglobulin A has been also found in our saliva, which also is responsible for neutralization of different pathogenic agents. So it can also act as an antiseptic agent for us, and it also helps us. It also helps us in the process of digestion. So we have seen that the process of digestion starts in our mouth, and it is the carbohydrates whose digestion starts in the mouth. No protein has been digested here, no lipid has been digested here, no nucleic acid has been digested here. However, we have mechanically broken down every type of the food, whether it was containing carb, whether it was containing nucleic acid, whether it was containing lipid or any other type of the biomolecule. With this, I am concluding my today's lecture. I was in a hurry because of some work in the market, so there might be some mistakes. I am sorry for that if some mistakes will be lying in my today's lecture, if I would not have been in a position to make you understand well, but still I tried my level best. And if you like my lecture, I once again advise or request you to get my channel subscribed and shared and liked as well. Thanks to all of you.